Good.
Are there any questions?
You said you were going to send us the results from our tests that were performed?
You're talking about for the lab?
Yes.
Yeah, I'll send those results tomorrow. Let's see, you did the gram stain on Sunday, and I guess I'm going to say that you put it in your drawer and you haven't looked at it yet, and you'll look at it on Tuesday. Um, I stated that you had started the lactose fermentation test today, and so you'll get your results to that tomorrow. And then the gelatin you started last Tuesday, and that takes a week. So you'll get your results also on Tuesday. All right? Any questions? Let me see. I'll start the screen share. All right, can you see my screen?
Yes.
Yeah.
Okay, good. Let's go over to here. So we're a little ahead of schedule, and we'll probably finish the antibiotics today. I'm not sure what I'll do tomorrow, but maybe we'll move on ahead with host defenses 1. Oh, I know what I should do. I should tell you a story about the first time that they had. Remind me to do this at the end of the lecture or at the second part of the lesson for today, and that is, no, wait a minute, we're doing the lab today. So, anyways, just remind me to tell you the story about the first time they used penicillin G in the clinic. It won't be really anything I'm quizzing you on, but it'll tell you about the first time they had the experience of using an antibiotic in a person or people, because I'll talk about the first 2 cases. So that's what we can do, and we'll do lab 9 today in the lab for the second part. And I'm not sure what we'll do tomorrow. I can go ahead of schedule, but the thing is I've already got a review in there, but that's all right. We'll go slowly through antibiotics and then slowly through host defenses 1 and host defenses 2. And I'm deliberately going slowly through these because, well, you're going to be tested on them in the comprehensive final. And so I want to go through and make sure that you'll understand something. And if you have any questions, please ask. So we were talking about antibiotics, and we talked about how they're using differences in the properties of bacteria compared to the properties of eukaryotes. And we haven't really talked much about that, but we'll talk a little bit more about it. And we did mention that with the penicillin family, it's taking advantage of the fact that bacteria have peptidoglycan and eukaryotes don't. And the penicillin family stops the ability of the peptidoglycan to form the crosslinks in the cell wall, and that puts a hole in the cell wall, and that gives rise to the cells bursting when water comes into the cell. And when bacteria are in your cells, or in your blood, whatever you want to call it, they tend to be in a hypotonic environment because they have more solutes in their cells than we have in our cells. And so that's why antibiotics work. And that is that water moves into them. I shouldn't say that's why antibiotics work. That's why members of the penicillin family work. And when they infect you, water moves into their cells, and if you can take penicillin, it stops the cross-links from forming, and then their cell walls don't give them protection, and their cytoplasm protrudes out of the holes in the cell wall, and that's why penicillin G works. Now, this has to happen when the cell is actively putting down its cell wall, and the penicillin family of antibiotics works best when the bacteria is actively growing. So if you give another antibiotic that slows down the growth of the bacteria, the penicillin won't work as well. Is that clear? We'll talk a little bit more about that, but just doing a little bit of review here. And the active ingredient for the penicillin family is the beta-lactam ring, this ring here, which is actually found, the beta-lactam ring, also on another family, the cephalosporins. And it's a totally different antibiotic, but it still has the beta-lactam ring. These are members of the penicillin family, and that's a cephalosporin. And the antibiotics can act narrow spectrum, and that is they act only on a certain group of the prokaryotes or the bacteria, like polymyxin B would definitely be a narrow spectrum antibiotic. And then there's ones that are broad spectrum where they work on more than one group, like tetracycline works on both the gram-negatives and the gram-positives and the chlamydias. And so that we call a broad spectrum antibiotic. And occasionally you get an antibiotic which you say, what is something like niazid, working on the mycobacteria and some of the gram-negatives. And we're not going to just address that question, okay? Everyone okay with that? And then when you're deciding on which drug to give a patient, like which antibiotic, you have to make a number of decisions for which one to do. And the decisions make the administrative route. Patients generally like to take a topical treatment of antibiotic or an oral treatment. Most adults don't like enemas. Or most people don't like IV or IM also because it's a shot. But the clinician has to consider how long they stay in the system, what the side effects are, before they decide on which antibiotic to give. All right, any question about that? So I don't think I mentioned terribly much, but the penicillins inhibit the cell wall, and there's a whole group of antibiotics that actually inhibit the cell wall synthesis, most of them stopping the formation of peptidoglycan. The cephalosporins, which is a family of antibiotics. There's about 70 members in that family, and that's more than the penicillins. There's, something like 40 or 48 members of the penicillin family. But also bacitracin, another antibiotic, works on inhibiting the cell wall. Vancomycin. Bacitracin is mostly used as a topical treatment of antibiotic, so you rub it on a cream. It's active. It is actually the active ingredient in bacitracin. If you've ever taken that antibiotic, it's effective against gram-positive bacteria, but it's not effective against gram-negatives. Vancomycin is rarely used. It's relatively toxic. It's mainly used as a backup antibiotic against a strain that is showing multi-drug resistance, like multi-drug resistant Staphylococcus aureus, MRSA. Vancomycin is the antibiotic that is what do you call it? The antibiotic of choice for MRSA. Okay, any questions about that? Either vancomycin, I don't remember, or chloramphenicol, which is a later drug I'll talk about. One of them, they found that it's less toxic if you highly purify it. So some of the toxicity came from trace contaminants in the antibiotic, but it is still toxic even when it's highly purified. Meaning there's severe side effects in something like 1 in 100 cases. And 1 in 100 is awfully high for severe side effects in an antibiotic. Any questions about any of that? I actually learned about that from one of my students who was apparently in the clinic, and he was in a clinic where they were using vancomycin and chloramphenicol. And like I said, when it's highly purified, it's less toxic, but it's still toxic even when it's highly purified. All right, so that's the first mode of action we talked about, drugs that inhibit the cell wall. Another mode of action for antibiotics is where the antimicrobial targets RNA transcription or DNA replication. The only antibiotics I'm briefly going to talk about are rifampin, which inhibits RNA transcription, and the quinolones and DNA nucleotides, which stop DNA replication. And we mentioned DNA nucleotides. It's a nucleotide which the antibiotic puts into their DNA, and it's not one of the 4 bases of DNA, one of the 4 nucleotides. And then the quinolones also are taken up by DNA replication, and they just stop DNA replication. Rifampin, how it actually works is the drug binds to the RNA polymerase, stopping its ability to bind to DNA and stopping transcription. Any questions about that? All right, so that's the mode of action where the antimicrobial either targets RNA transcription or DNA replication. A big area of antibiotics is where the antibiotic stops protein translation. And as you recall, protein translation of prokaryotes and eukaryotes is a little different. The most obvious difference is that the ribosome is different. The ribosomes for the prokaryotes is 70S, and the ribosomes for eukaryotes is 80S. And because of the differences in protein translation in the 2 cells, a number of antimicrobials have been developed which stop protein translation. Any questions about that? So there's a lot of them that work on stopping protein translation. And do you guys remember I briefly mentioned that we don't worry about the archaea because none of them are human pathogens? But if an archaea were to ever develop becoming a human pathogen, then we will be in trouble because most of our bacterials and most of our antibiotics, they don't work on the archaea. They only work on the bacteria. Remember I talked about that? Well, the few antibiotics that actually do work on the archaea belong to this group of stopping protein translation. And if you think about it, it's not that surprising because the ribosomes of archaea are similar to the ribosomes of bacteria, much more similar in that respect than they are to the ribosomes of eukaryotes. Okay, so this is just a side note, and that is the few antibiotics that do work on archaea tend to be antibiotics that stop protein translation. And we'll talk about each of these in a little bit, but you can have an antibiotic actually interfering with the TNA's ability to read the messenger RNA, and so that gets read incorrectly, and that can be lethal to the cells because protein translation won't happen correctly. You can have an antibiotic binding to the ribosome. It's actually the ribosomal RNA, but it's the ribosome. And then that inhibits the formation of the peptides being linked on the growing chain to the new peptide on the new tRNA, or, yeah, that's right, tRNA. And so that stops protein translation. You can have the antibiotic binding to the ribosome and then preventing it moving along the messenger RNA. And we'll talk about each of these. So erythromycin does that. And then tetracycline actually interferes with the tRNA anticodon reading of the messenger RNA, so that's similar to streptomycin. And those are different ways you can foul up protein translation. The first family of antibiotics that stop protein translation is the family of tetracycline. It interferes with the binding of tRNA to the messenger RNA ribosome complex, and the messenger RNA is not read correctly. Tetracycline is a very broad spectrum antibiotic, meaning it hits both the gram-negatives and the gram-positives. This is actually a problem because that means tetracycline will kill the normal flora of the gastrointestinal tract, and patients may get an upset stomach or diarrhea or constipation. It's kind of funny that antibiotics sometimes give side effects where somebody gets constipated and other people get diarrhea. Tetracycline, like I said, is a broad spectrum drug, so it can give other side effects, like the normal flora can be knocked down on the other parts of your body, such as in the vagina flora, and that may give rise to, you're not seeing it there, a super infection with the yeast, Candida albicans, or you can get a skin infection because the normal bacteria on your skin are knocked down. And then something that's a little different, meaning it's not responsible for the killing off the normal microbiome flora. Tetracycline, actually, when it's given to children, causes them to get discolored teeth. Like this is actually an adult, you can tell from the teeth, but this adult was given tetracycline as a child, and it causes discoloring of the teeth. See the bands here? That's a side effect that happens with tetracycline when it's given to children. So tetracycline is not usually given to children. I think that has to be young children, not children like 13. And then tetracycline also can cause liver damage in pregnant women. So those are some of the side effects with tetracycline. Fortunately, the liver damage is pretty rare, and also the yeast infection bloom is also fairly rare, although the upsetting the stomach and giving diarrhea is much more common. I mean, greater than 1% of the patients get that, much greater, probably greater than 10% of the patients get that. And then, like I said, the staining of the teeth in children is also quite common. My nephew actually took tetracycline as a young child, and he kept getting ear infections. And the doctor told him and his mother, who took him into the doctor when he had an ear infection, that he was a young child when he got the ear infection. And the doctor said, okay, I'm going to give you tetracycline this time, and that's going to stain your teeth. But the next time you get an ear infection, you're going to have surgery and get the tube put down the throat. And that scared him so badly, he didn't want surgery, that he actually never got an ear infection after that. Any question about that? But he does have the stained teeth because he took tetracycline as a young child. If you don't know, young children, their connection from the ear to the throat, it's fairly straight. In an adult, it goes down. And so in young children, they're much more likely to get that connection between the ear and the throat plugged when they're sick, and that causes ear infections. Any questions about any of that? All right, erythromycin, another antibiotic which stops protein translation in the bacteria, but not in the eukaryote cells. It binds to the ribosome and keeps the ribosome from moving along the messenger RNA. Erythromycin is not effective on many gram-negatives. Notice how I said many, but it is effective on some. And so if you look at the chart here, when you ask people, is erythromycin broad spectrum or not, and actually it would be considered broad spectrum because it works on gram-positives and rickettsia, but it only works on many of the gram-negatives. It doesn't work on all of them. And so you can get an argument here, does erythromycin work on gram-negatives? And we're not going to get into that argument, so let's just avoid it. You just need to know that it's effective on some gram-negatives, but not all of them. So on some of the gram-negatives, why it doesn't get in them is because of that outer membrane of the gram-negative cell. At least some of the species of gram-negatives prevents the erythromycin from getting through that outer membrane. It is effective on gram-positives and some gram-negatives, and I do not know why it's effective on some gram-negatives and not on others. It must have to do with the difference of the protein channel in the outer membrane. All right, any questions about erythromycin? Okay, streptomycin was an antibiotic that was very common. Actually, erythromycin was also very common when I was a child. Both of these antibiotics aren't used as much as they used to be because of the development of drug resistance. But streptomycin still is effective against many species of mycobacteria, so it is a drug of choice. In fact, if the mycobacteria isn't susceptible to streptomycin, that is one of the drugs, meaning the first choice the doctor will use against mycobacteria. It is effective on gram-negatives as well, so it's a broad spectrum drug. As I stated, this was very common in the 1960s and 70s, but because of the development of drug resistance, it's not used as commonly as it was in the past. Okay, any questions about that? Let me state that when you're treating mycobacteria, these cells grow slowly, So the drugs have to be used for a long period of time. I'm talking about for months. And they don't usually use just one antibiotic to treat mycobacterial infections. They will use a cocktail of something like 4 drugs. But streptomycin is one of the preferred antibiotics in the cocktail for mycobacteria. The reason is that a lot of antibiotics will not go through the cell wall of mycobacteria because it has so many lipids. It has mycolic acid in the cell wall. But streptomycin is an antibiotic that will go right through the mycolic acid in the mycobacteria. And there are a few that do that. And those are the ones used against mycobacteria. All right, any questions about that? Maybe that's why it's used on gram-negatives. The streptomycin goes right through the outer membrane of the gram-negative cell. Chloramphenicol binds to the ribosome and prevents it from linking the amino acids together, stopping the peptide bonds from forming. And that, of course, stops protein synthesis. And it's broad spectrum. It works on the gram-negatives and the gram-positives and also the archaea bacteria, but we won't talk about them. But chloramphenicol, even though it is a very good antibiotic, it has severe side effects, so it's not used if something else is available. If there's another antibiotic that can do the trick, they'll use that instead of chloramphenicol. Some of the severe side effects is it can cause potentially fatal anemia in some patients, and also fatal neurologic damage, or even any neurologic damage is bad. Okay? All right, any questions about chloramphenicol? So those are 4 antibiotics that stop protein translation. All right, if there's no questions about protein translation, let's move on to the inhibition of a metabolic pathway, meaning this is the mode of action. The antibiotic stops a metabolic pathway. And usually that means that the microbe has this metabolic pathway and the eukaryotic cell does not. Trimethylprim and the sulfa drugs is one that I'm going to talk about. And let me see if I can bring out a new thing here. Let's see if I can do this. PABA, para-amino benzoic acid. And we have a drug. You know, I'm going to have a hard time doing this, so let me go here, and I've got it already done in the long notes. There it is. So let me open it up here. Here it is. So you don't need to know that PABA is para-amino benzoic acid. That's a molecule in the bacteria cell, and the sulfa drug prevents PABA from changing to dihydrofolic acid. Let's see if I can blow that up a little. The spacing's a little off, but that's okay. This is a B vitamin, so is trihydrofolic acid. Trimethylprim stops the switching of the dihydrofolic acid to the trihydrofolic acid. And then the bacteria cell can take trihydrofolic acid and convert it into a nucleotide, and then take this nucleotide to build DNA or RNA. This pathway does not happen in in eukaryotic cells. We build up our nucleotides either from getting the nucleotide in the diet, meaning we're eating a little bit of DNA, or we can take the B vitamin, the trihydrofolic acid, and convert it into a nucleotide. But we don't start from the molecule PABA, okay? But the bacteria cell does. Any question about that? So they can build up DNA from PABA. And the sulfa drugs stop the first reaction, switching PABA to DHF, and trimethylprim stops the second reaction from DHF to THF. Anyways, they can stop this pathway in the bacteria, and then that will be lethal to the bacteria because they can't make their DNA, and it doesn't harm the eukaryotic cell. And that's the way trimethylprim and the sulfa drugs work on bacteria cells. Any question about that? Now, one way the bacteria cell can evolve drug resistance is they can get rid of this pathway. They can kill your cells or take trihydrofolic acid from your blood, because this is a B vitamin, and then build the nucleotide that way. Or get the nucleotide from your dead cells and then build their DNA. And sometimes that is how bacteria evolve drug resistance to either sulfa drugs or trimethylprim, or perhaps even to both of them. Just abandon this pathway. There we go. Folic acid, which is dihydrofolic acid, and trihydrofolic acid is actually vitamin B9. And we can build our nucleotides from that. But like I said, we also get most of our nucleotides directly in the diet. Whenever you eat a cell, like especially in meat, you're going to get some DNA. And that's true for all cells, like watermelon. You get a little bit of nucleotide when you eat the watermelon because there are nucleotides in the plant cells, meaning DNA in the plant cells. There's also RNA, which we can digest that too. All right, any questions about that? Let me state that when trimethylprim and a sulfa drug are used, they're usually used together in combination because the two drugs act synergistically, meaning that if you use one, it will work better when the other one is around, meaning the two work better together than they do by themselves. And how you can look at that synergistically is, this is a Kirby-Bayer test, and let's say this is trimethylprim. And when you put it down, you get a zone of inhibition like this, where the dotted line is. Everyone see that? And I can maybe blow this up a little bit. So the white cloud is where the bacteria is, and the dark region is where there's no antibiotics. And this would be, let's see, I said that's trimethylprim. So this would be one of the sulfa drugs, and I don't know what that is there, so we'll just look at this and ignore that. And this region here would be the zone of inhibition, the dotted line, for the sulfa drugs. And when you put both of them together on a plate, you have more than just the zone of inhibition for trimethylprim and the zone of inhibition for the sulfa drugs. You also have a zone of inhibition inside this green area, which happens because the two drugs work synergistically, meaning that in this region here, because you have both of them here, it's preventing the growth synergistically. Any questions about that? So the zone of inhibition is greater together than either would be by itself. The two drugs work synergistically, meaning they work better together than by themselves. Any questions about that? And there are some antibiotics that do work that way, synergistically. That's a little too small. And trimethoprim and the sulfa drugs work synergistically. There are other antibiotics that sometimes work synergistically together. Now, if two drugs work worse when both of them are applied at the same time, then they are antagonistic. The two inhibit each other. And so instead of seeing the normal zone of inhibition, the zone of inhibition will be smaller if the two are antagonistic for each other. With penicillin, I mentioned that that drug is antagonized by another antibiotic that slows the growth of the bacteria. And so that first antibiotic might not be antagonized by penicillin, but this one, because it's near this one, can actually make the penicillin zone of inhibition less, because this antibiotic slows the growth of these bacteria. And that's when they're antagonistic. Okay? Any question about that? Any questions about the mode of action of antibiotics? This would have been a really good thing for you to study in the lab, but unfortunately we can't do that because, like I said, frequently when you're applying penicillin with another antibiotic, you see the other antibiotic, there's about 50% of the other antibiotics slow down or reduce the size of the zone of inhibition, at least in this region, for penicillin. Finding two that work synergistically, like trimethoprim and the sulfa drugs, that's more rare. Only a few antibiotics work together synergistically. All right. Any questions about anything? If not, we've talked about antibiotics and how they inhibit bacterial cells, and we've been very good at developing antibiotics because there are differences between the cells of eukaryotes and the bacteria. And because there are differences, we've developed drugs which take advantage of these differences, and that causes selective toxicity where the bacteria cell is being killed or inhibited, and the eukaryote cells are left pretty much alone. Now, we don't have any antibiotic which is 100% selectively toxic, and the main reason for that is that we have a lot of bacteria as part of our normal microbiota, and whenever you take antibiotics, you're going to disrupt the normal microbiota. Okay? So there's none that's 100% selectively toxic, but there are some which are fairly selectively toxic. And we've just discussed a lot of them. We talked about the penicillin G and the penicillin family in great detail. And they're selectively toxic because they're hitting peptidoglycan and they're leaving the eukaryote cells alone. So because of that, we've developed a large number of antibiotics that work against the bacteria. But what about other pathogens? So we've talked about antibiotics and they're stopping bacterial infections, but there's other cells which cause infections, such as viruses, protists, which are eukaryotic cells, worms, which are animals, obviously eukaryotic cells, yeast infections, which are also eukaryotic cells. And we don't have any infections from the archaea, so we're not going to talk about them. But there are human pathogens, And we have developed drugs to stop each of these other human pathogens from infecting humans, but none of them have developed the selective toxicity that we have with the antibiotics that work against the bacteria. So we've developed drugs like against viruses and against protists and against worms, but they haven't been as selectively toxic as the antibiotics. And my question for you is, why do you think that might be? Come on. This is an important point. I could give you the answer, but I guys want you guys to think. So come on. Just start thinking. Let's talk among yourselves. What's different between the bacteria and these other human pathogens? Obviously, they're not bacteria. That's one difference. What else?
Complexity?
They are more complex, yes. Although the viruses are simpler. So why are the viruses... Say again?
They don't need a host to replicate.
Yeah, the viruses need your cells to replicate in. So obviously, you're looking for a drug that will hit the virus but not hit your cells. And the viruses are replicating in your cells. So that's pretty hard to find one that's selectively toxic on the virus and not going to be selectively toxic on your cells. So certainly for the viruses, that explains it. And that is a virus, it's infecting your cells. So that is your cell already. Okay? So it's harder to find a drug that would be selectively toxic to the virus since it's replicating in your cell. In the bacteria cell, it's really different from your cells. Where the virus is infecting your cell. So it's harder finding a drug that's selectively toxic for a virus. Okay, what about the protists and the worms and the yeast? Why has it been harder to find a drug that's selectively toxic on those cells? I'll give you a hint. Remember this table here? Nobody wants to comment?
So the lack of a peptidoglycan wall.
Yeah, the bacteria have differences from the eukaryotes, and the reason why we've been so successful on developing antibiotics is because they're taking advantage of the differences between the bacteria and the eukaryotes. All of the other cells that are human pathogens are eukaryotes. And so they're not going to have great differences between our cells and the human pathogen. And so it's been harder to find a drug that is selectively toxic because there's less differences between those cells and our cells. A yeast cell that infects us is a eukaryote, the same as our cells. Okay? And that's why we've been less successful in developing selectively toxic drugs for the other human pathogens. Because there's less differences. Okay? Everyone see that? All right. If there's no questions, we'll stop here and we'll start at 2:30 in the lab.
Yeah, quick question.
Okay?
Since viruses need living host cells to replicate, how do they culture outside of the body for being able to do tests?
How do they culture viruses outside of the body? Is that your question? Sorry, I'm getting an echo here. Maybe I've got the volume on too loud. Let's reduce this. Nope, that's not loud at all. You can grow human viruses in tissue culture in human cells. Okay? And then you can grow the viruses in the human cells in tissue culture. So that's generally the way they do it. With some viruses, you can find another animal that they grow in. There aren't a whole lot, but they can grow HIV in one of the chimpanzees. It doesn't cause AIDS in chimpanzees, but the virus will grow in the one of the chimps. But mostly viruses, at least human viruses, are grown in tissue culture. And you just find the cell that will grow in, and if you can grow that cell in tissue culture, you can usually grow the virus in that cell. Okay? All right. If there's no questions, I'll see you at 2:30.